Hello. Sorry, we're running a little bit late. I'm coming from another class, so give me a few seconds to set up. Okay, thanks for your patience. So before we get into our Mentimeter session, I just wanted to highlight a few things. So I noticed that the guest speaker had a bit more advanced diagram showing a lot of intermediates. And so I thought I'll show a simple one, a more simplified one, looking at the role of vitamin A in gene expression. And so as we saw yesterday, vitamin A has a role that it plays in gene expression. And if you recall, this diagram looks very much like what we'll be seeing for vitamin D. We did see it yesterday for vitamin D, but I just wanted to talk about those two functions again. Okay. So we just want to look at how vitamin A plays a role in gene expression. So what vitamin A does is that it binds to a protein receptor. And by binding to a protein receptor, we have a vitamin protein receptor complex, right? And it is this complex that in turn will bind to a regulatory region in the DNA, and then the gene is expressed. So if you recall from proteins, we talked about how genes are expressed, and very often you have messenger RNA lining up or pairing up on the gene, the DNA material itself, and then copying that message, and then it's able to translate it, and then a protein is produced. So vitamin A plays an important role in how genes are expressed. And it does that by binding to what we call a protein receptor. The protein receptor and the vitamin become what we call a ligand, or you can call them a vitamin protein complex. And then they do copy that information, and then it will go through that process of translation till we get a protein. So this is listed up here. First of all, we have retinoic acid, which is a form of vitamin A, as we saw yesterday. It goes into the nucleus, and then it binds to a protein receptor. So now you have the vitamin and protein receptor binding to a regulatory region of the DNA. It is transcribed. So transcription of the gene is turned on, and it increases the amount of this messenger RNA that is made. And so it is this messenger RNA that directs translation. So if you recall what translation is, Essentially, what you have is transfer RNA also coming to pair up with this messenger RNA. So please take a look at your protein synthesis slides again. You have a transfer RNA now pairing up with mRNA with the help of a ribosome. And it's at this point where the gene is translated and then a protein is expressed or a protein is produced from this coding of the DNA. And so that is how vitamin A will play that important role, okay? So what you have, so what happens without vitamin A is that this transcription process is impaired because you do not have enough to form the ligand that we're talking about, which is the same as the vitamin protein receptor for all of these processes to take place. I also wanted to talk a little bit about a similar function that we see for vitamin D. And so almost a similar diagram. Vitamin D also functions in gene expression, and we've already seen this figure, and it's exactly the same as the figure for vitamin A. So here again, we see vitamin D also acting as a ligand, helping with the expression of a specific gene. And we did see how vitamin D helps through this process to produce the calcium-binding protein. And so that is the protein that vitamin D helps with this expression. And so here we see an enterocyte or a mucosal cell. We see vitamin D through its process acting as the ligand, and then forming that vitamin D protein complex. What you have being produced, or the protein that is being expressed, is the calcium transport protein, which helps with absorption of calcium, right? And so we see how vitamin A and vitamin D play a role as ligands. I just wanted to point that out because the diagram for vitamin A yesterday was a bit complicated. Okay. So what we'll now do is to go into our Mentimeters session. Give me a second to turn that up. And then we can try to answer some questions, first of all, on our water-soluble vitamins that we talked about yesterday, and then also about the week before where we looked at weight management and obesity. Bear with me, give me a second. Okay, so this is so we can go to menti.com and use that code to access the Mentimeter question session. Let me just make sure I'm at the beginning of the slides so we can go through all of them. So our first question is, by what mechanism does vitamin A deficiency cause xerophthalmia? So we're just going to wait till we see a few more people participate here, and then we can turn it on to see what people think. Okay, so we're just going to show the responses. So we have most people saying that it prevents the differentiation of newly formed epithelial cells into mucus secreting cells. That is the popular vote. And then we have a few people saying by preventing the differentiation of keratin containing cells into antibody secreting cells. Oops. So I wanted to show us the correct answer. So the correct answer will be preventing the differentiation of newly formed epithelial cells into mucus secreting cells. Because we did see that vitamin A plays a role in the differentiation of cells, if you recall from yesterday. And so when we have xerophthalmia, if you recall, there was a diagram with an eye that had a whitish substance on it. And so we see the cornea being blocked, and the cells are literally being changed into keratin cells. So keratin cells are what we will have in, for example, our nails. And so without vitamin A, now instead of having the epithelial cells here being differentiated into mucus secreting cells, which is typical of epithelial cells, you have them being differentiated into keratin cells. And it is the keratin that clouds the eye in xerophthalmia. Okay. So that will be the response for this one. Next question, it says a patient has elevated blood homocysteine levels despite him having adequate protein intake. So based on the homocysteine hypothesis that we saw yesterday when we're looking at vitamin Bs, which intervention is, so by saying that I have given a clue. Which intervention is most appropriate to reduce cardiovascular disease risk that is related to having high levels of homocysteine? What do we think? So maybe we can have a few more people signing as well. Okay, so let's see, because we don't have a lot of time, let's see. Okay, so we have most people saying supplement with vitamin B6, B12, and folate, and that is the correct response. Why is this correct? Yesterday we looked at the homocysteine hypothesis, and we did see how these three vitamin Bs, vitamin B6, B12, and folate, and he did mention that folate is also vitamin B9. And so these three play a role in regulating how much homocysteine that we have in our system. And so very often high homocysteine levels has been linked with increased risk of cardiovascular diseases. So which statement does not describe a B vitamin? Which of the statements below? Okay, let's show the responses. Okay, so this is a pretty divided one. Okay, so the correct response is a ligand for a nuclear receptor controlling cell differentiation. So we just looked at vitamin A and vitamin D that play a role as a ligand. So we talked about how the ligand is the vitamin D or vitamin A going to attach to a protein receptor, and then it becomes a transcription factor leading to transcription of a gene, leading to its expression after it's been translated. So vitamin Bs do not play a role in this. So we saw vitamin Bs playing a role in homocysteine regulation. They do play a role in energy metabolism as well, but they do not act as ligands. Okay. A coenzyme in metabolic pathway that converts amino acid to energy. We did see this as well. If you recall yesterday how it plays a role in deamination in other different pathways, and that will be for vitamin B. It's a coenzyme in the metabolic pathway that converts fatty acid to energy. That is correct. And the first one says it's a vitamin that reduces the risk of spina bifida. That is wrong because we know it's folate. Well, that is a wrong response because folate is a B vitamin and it does that, right? Folate is responsible for neural tube defects, and spina bifida is an example of that. Okay, so let's try this one. What is the difference between megaloblastic anemia and pernicious anemia? If you have any follow-up questions, you can just let us know and that we can answer as we move on. Okay, so let's see the responses here. What is the correct response? The correct response is the first one. So if you recall, there's no difference when it comes to the symptoms between the two anemias, but one is caused by an autoimmune response, right? Megaloblastic anemia is characterized by abnormal cells called megaloblasts, while pernicious anemia is characterized by the presence of macrocytes. So you can have macrocytic even when with megaloblastic anemia as well. Remember, the megaloblastic is that big cell that doesn't divide, but it still has a nucleus, and the macrocytic is the one, it is still a big-sized red blood cell, but the nucleus is missing, if you remember from that diagram from yesterday. Megaloblastic anemia is caused by a deficiency of folate, while pernicious anemia is caused by a deficiency of vitamin B12. Not completely correct, and there's no difference between the two anemias. Symptomatically, there's no difference, but the causes are a bit different. So which statement best describes the role of micronutrients in gene expression? So this question is really looking at different types of vitamin A. Okay, let's see what people are thinking, and then let's show the correct answer. So both retinoic acid and vitamin D influence the transcription of DNA into messenger RNA. So if you remember yesterday, we talked about retinol, retinal, and retinoic acid, which are all different forms of vitamin A, but the form that is involved in gene expression is the retinoic acid. And we've already seen how they are both involved as ligands helping with transcription. Okay, so let's look at this question. Food fortification with folic acid has significantly reduced neural tube defects in Canada. However, concern exists about excessive intake. Which scenario best reflects the evidence presented from yesterday? So essentially saying we do know that it's helpful in reducing the cases of neural tube defects in Canada, but what could be other concerns related to high folic acid intake that comes from the fact that our staples are supplemented or they are fortified? So maybe wait for a few more people to respond and then. Okay, so let's show the responses. And let's show the correct answer. So the correct answer is that high folate intake may reduce cancer risk unless in cases where you have pre-existing cancers. And so if you recall, we did see a diagram where there was a bump after the year, a bump like an increase in number of cases of cancer after the year that supplementation started in Canada. But after a while, that bump reduced and it came back to the same levels before the introduction of fortification of folate. And the explanation was that they think the bump is as a result of people who already had pre-existing cancerous cells. And what happened with the supplementation or the fortification was that now they were suddenly exposed to a lot of folic acid, which helped with the growth of the cancerous cells, increasing, that cancer levels. But after a while, we can see that the levels of cancer reduced again when we looked at that diagram. So what happens is if you already have some cancerous cells, folic acid could, enhance that and make, move you into having more cancer cells because there'll be rapid growth of that. However, if you do not have it, it can be, if you do not have pre-existing cancer, it can be protective. What is not a function of vitamin D? Which of these ones is not a function of vitamin D? So maybe just a few more people. Okay, so let's show the responses. And let's see the correct answer. Okay. It increases urinary excretion of calcium. So that is not a function of vitamin D. Vitamin D helps maintain blood calcium levels. We talked about how vitamin D helps with the gene expression of the calcium binding protein, which in turn helps with the absorption of more calcium into the blood. But it doesn't have a direct control in increasing how much calcium is excreted in the urine. Okay, so next one, our question is about why are plant sources of vitamin A unlikely to cause vitamin A toxicity compared to animal sources? And so if you recall yesterday, we talked about, just a little clue, we talked about preformed vitamin A and provitamin A and the differences between the two. Just a few more responses. Okay, so let's show the answers. So plant sources of vitamin A are unlikely to cause vitamin A toxicity because we know beta carotene conversion to retinol is inefficient and regulated. So if you recall, we talked about preformed vitamin A, which is retinol, and then we talked about well, I didn't talk about it, but the guest speaker talked about provitamin A, which is really from plant sources, so the beta carotenes. And then other forms of vitamin A that do not have vitamin A activity, but serve as antioxidants in the body as well. Okay. And so we talked about retinol activity equivalence, RAE, and we talked about how one retinol activity equivalent is equal to 12 micrograms of beta carotene. And there were some reasons why this conversion is inefficient, because the body will regulate how much it absorbs. And so because it's regulated, so well regulated, you cannot go into toxicity. Very rare to go into toxicity from just plant sources. However, animal sources have the preformed type, retinol, and especially if you're consuming it from sources such as liver, which are concentrated in vitamin A. One, if you eat it very often and often going over the UL, you could run into toxicity with that. Right? But the body is efficient in making sure it absorbs what it needs and tries to balance that vitamin A level in the body. And one of the worst sources that can run one into toxicity is through supplementation. Okay, so next question is about, and sometimes I do have to cut the question because they are not enough. It doesn't allow me to put the whole question there because of the limited characters. So observational studies suggested that beta carotene intake was associated with reduced lung cancer risk, whereas interventional trials using beta carotene supplements showed increased lung cancer risk in smokers. Which explanation given below best explains why this is? Just a few more responses. Okay, so let's show it, the responses now. And let's look at the correct answer, okay? So the correct response here is that observational studies examined beta carotene consumed as part of whole foods. So typically in observational studies, you're collecting information, perhaps with food frequency questionnaires. This is people's typical diets, and vitamin A intake is assessed from that. So it's part of what they eat and not usually from supplements, right? In intervention trials, very often you're given the intervention and supplements are provided for a group of people, and they are compared to the control who are not given those supplements. And so it was the high supplemental doses, very often in the preformed type of vitamin A, that led to some of these increase in cancer risk in smokers, lung cancer risk in smokers. Okay? So this is kind of related to the question we had before as well. So a patient consumes high doses of folic acid supplements, and then we have tests. When tests were done, it shows normal red blood cell size, but the patient reports of tingling and balancing issues. Sorry, I had to cut the question. So what is the most likely explanation? What is going on here? Okay, so let's see what people have. Okay, so we had quite a number of people talking about folate toxicity. But if you recall from yesterday, we talked about vitamin B12 deficiency leading to neurological damage. Okay. So we talked about vitamin B12 deficiency leading to neurological damage as well. And we talked about how when you have excess intake of folic acid supplements, and that's why vitamin folate UL, the upper tolerable intake, is capped at 1,000 micrograms, so that you cannot go over that, because once you go over those levels, you're at risk of having your vitamin B12 deficiency being masked, right? And so what happens is that there is still some, what we typically see is that although you might not see the cells, the red blood cells changing in their size, you will have that neurological damage as a result of vitamin B12 deficiency still going on. However, when they look at the blood cells, it will not show that you have anemia related to folic acid, right? And so the correct response is vitamin B12 deficiency being masked by folic acid. And so if you recall the case study that he showed us yesterday of Mr. Chen, and so Mr. Chen had a condition where he had atrophic gastritis, right? And so because of that, he wasn't producing enough intrinsic factor. And because he wasn't producing enough intrinsic factor, and this example is actually in the textbook, because he wasn't producing enough intrinsic factor, he wasn't able to absorb his vitamin B12 optimally. And one of the symptoms that he had was tingling and balance issues. So neurological problems as a result of the vitamin B12 deficiency that was happening. Okay, so next question. A public health researcher is evaluating Canada's folic acid fortification program. Which population is at greatest risk of harm if folic acid, give me a second. This question is incomplete because it cut short. So a public health researcher is evaluating Canada's folic acid fortification program. Which population shown below is at greatest risk of harm if folic acid intake is high? Sorry, there wasn't enough characters and I couldn't put it up there. And please note greatest risk of harm, meaning that there may be some groups that may be harmed, but which of these population groups will be at greatest risk of harm out of all that we see? And it is related to some of the things that we've already discussed, some of the questions that we looked at. Okay, so let's see responses. Okay, so the correct response is C. Why is it not B? It's not B because pregnant women not taking prenatal supplements are actually the best to be helped if folic acid intake levels are high, right? Because women in their reproductive ages usually have higher needs and that need to be met so we can avoid neural tube defects, okay? However, in older adults, when the folic acid intake is high and they have impaired intrinsic factor production, then they're at a higher risk of vitamin B12 deficiency being maxed, right? And so they're at a higher risk of neurological damage, the tingling and the balance issues that we talked about, because there's so much, they're consuming so much folic acid intake, and their intrinsic factor, which is important for absorption of vitamin B12, is impaired, right? So young adults consuming plant-based diets so they are at some risk of harm if their vitamin B12 is not high enough, because we know vitamin B12 are usually sourced from animal foods, right? So you have higher consumption of vitamin B12 if you're consuming animal foods. But then with regards to the immediate harm, but then they still have their intrinsic factor intact in young adults. And so at least we know the absorption of vitamin B12 from whatever plant-based diet they're eating is not limited, because their intrinsic factor is intact. The production of that is intact. And so the group with the highest harm will be this group. So children, non-vegan, consuming fortified cereals, they're non-vegan. We know that their vitamin B12 is at optimal levels. So we're not worried about vitamin B12 deficiency for them. And so the group at highest risk will be the older adults, because even if there's vitamin B12 in the food, it will not be absorbed because of their intrinsic factor production. Okay? So give me a second. I'm going to transition to our obesity questions, and then we can review that as well. I think it's this one here. Okay. Okay, so which of the following is a characteristic of an obesogenic environment? We talked about this. Please keep in mind the code is different. For some reason, I'm not able to show the responses. So what do we think? Because I'm not able to show your responses for, not sure why. Maybe there's a glitch in it. So let's discuss it then. If tech fails, we go back to the old ways. Which of the following is? Yes, go ahead. A, so readily available fast foods will be a characteristic of an obesogenic environment, because we talked about them being environments that help promote weight gain, so help promote sedentary lifestyles and eating foods that are high in saturated fats, salts, and sugars, free sugars. And so all the other examples, walkable neighborhoods promote higher physical activity, workplace gyms, higher physical activity, public transit, you're forced to walk a bit. So that is also helpful for a leptogenic environment. Okay. So what statement about BMI is correct? If person A, so there is a little text here that involves a little bit of calculation. So person A has a weight of 65 kilograms. Oh, it's working now? Perfect, okay. So person A has a weight of 65 kilograms and a height of 165 centimeters. And then person B has a weight of 65 kg and height of 175 centimeters. Which statement about their BMI is correct? And I'm just going to move through this a little quickly so we can answer a number of questions before the time ends. Okay. So person B has a lower BMI than person A. That's what most people are saying. Then person A has a lower BMI than person B. So in calculating the BMI, remember your BMI is equal to your kilogram per height in meters squared. So you have to convert the centimeters to meters before you do the calculation. Okay. Which of the following is a limitation of BMI? And I'm just going to leave the responses on. I think it glitched again. So what do we think the correct response is? If tech fails, we go back to, I'm not sure why it keeps turning on and off. So what would be the correct response here for the limitation of BMI? Yes. C, it cannot estimate visceral fat. So if you recall, we talked about visceral fat being the fat that is around your organs, and very often is linked with central obesity. So having fat around the waist region, right? And so how the fat is distributed plays an important role. And BMI is not able to estimate visceral fat efficiently. And that's why we talked about at individual level when we're using BMI to understand someone's risk of some of these comorbidities, it's important to measure waist circumference as well to determine their risk. Which is the best dietary pattern for treatment of obesity? What do we think? I don't know if it's back. Okay, it's back. Okay. So I'll just wait for 2 more people because we're at the end. Okay. So the correct response is a nutritious diet that matches patient preferences. If you recall, we talked about how a diet that, we talked about some features, important characteristics of a diet that is supportive of weight loss. It's important that it is sustainable and it meets the dietary preferences of the person, at the same time making healthy choices the easiest choice. And then offering support with a health professional. So with respect to the 5A approach, what A is represented by, is it all right if we discuss your weight? So if you remember, we talked about the guidelines to support treatment of obesity by Canada, by Obesity Canada. And ASK was the first one of the 5As. Okay. So after cognitive behavioral therapy, which is one of the management tools for obesity, Andrew learned to substitute carrot sticks for the jelly beans he used to eat while studying. What is the antecedent? Antecedent, I'm saying it right. What is the antecedent? So correct response, studying is the antecedent. We talked about how the antecedent is that activity that is taking place, and very often you have to identify that in CBT to know what is typically happening. And this helps us to tailor the intervention that's being provided by the nutrition professional, the health professional, to the needs of that person. And so the antecedent here is studying. So what is the mechanism of action of orlistat? So maybe this will be our last one because it's 2:59. Orlistat. We looked at orlistat, we looked at Ozempic, we talked about Mounjaro, a number of them. Okay, let's show the correct responses. So Orlistat, as a medication, what it does is it inhibits lipase